right, good morning, and thank you again for the introduction. And it's a pleasure to be here today and to tell you a little bit about part 2 of the talk that I gave yesterday, which was a lot about how can you design different types of biomaterial niches or systems with which you can culture cells in 3 dimensions. And the goal is to try to control the cellular microenvironment so that you can regenerate functional tissue structures. And we looked at some different examples of this. But inherently, many times when we attempt to grow these tissues, a lot of times the properties are suboptimal. You might regenerate cartilage that has half of the desired properties of articular cartilage. Or it becomes very, very difficult to get neurons to make functional synapses with surrounding muscle tissue. And so it makes us take a step back and to think about what's missing and how could we design better systems and what information might help us improve on this. And so part of this is really still our lack of understanding of how does a cell sense its environment, incorporate that environment into changes that could be useful towards tissue engineering. So today I'm going to work a little bit in this direction and say, how can we develop and use these types of systems to get at these fundamental biological questions? And this is going to be in the context that my group is really interested in using hydrogel scaffolds and different types of chemistries and ways to process and change those material properties. And I'm going to give you 2 examples today. One is how you can use light to tune the material properties and change it in dynamic ways. And by doing that, you can learn some important information about this signaling. And then the second part is that biology is complex, and many times it's not about a single signal, but it's about integrating multiple types of cues. And so oftentimes this means we need to look at what I'll call orthogonal chemistry. So how can you systematically make changes in controlled manners that might involve 2 or more functionalities? So with that context, I'd like to remind you a little bit about hydrogel systems. So here's a macroscopic image of a hydrogel. And again, a hydrogel, the molecular level details of this, if one could see it, would be long macromolecular chains that normally would dissolve in water. But because of various interactions between these chains, hydrophobic or covalent, it renders the material insoluble, and instead these types of systems imbibe large amounts of water. Now because of that, in that context, they become very nice environments for culturing cells and many primary cells, so cells that you get directly from a patient, in this hydrogel system. So the high water content imparts tissue-like elasticity for many of the cells that are found in environments that are soft. It also allows transport of many molecules that you might like to deliver from the media to the cells or that cells might secrete to one another, and is very important in their communication. So we're very interested in controlling this extracellular material matrix from the molecular level details all the way up to the macroscopic structure and processing, and being able to understand and manipulate that on various time scales, size scales, with spatiotemporal control provides a real good opportunity. And it's one that engineers have a lot of interest in analyzing, because many times you're trying to figure out rate limiting steps, the most important information, and scale up of these types of complex processes. So there are many, many different types of hydrogels, and they all have various advantages. But my group has had a lot of interest in PEG-based hydrogels. And from the applied side, it's because PEG, polyethylene glycol, is widely used in human medicine. But for today's example, from the fundamental side, the reason we like PEG hydrogels is because PEG is one of those few synthetic materials that prevents non-specific interaction of various proteins. So when you're culturing cells, you add a complex milieu of media that's filled with many different types of proteins and signaling molecules. And many times, these will just non-specifically adsorb to a surface or a substrate. And then the cells will sense those instead of your material. So PEG is desirable because it minimizes some of those non-specific interactions, so that if I want to introduce a cue and see how the cell responds to that particular cue, it provides a background that is a blank slate with which I can decorate these extracellular environments. So in a really simple sense, we take polyethylene glycol and we put different types of functional groups. Here's just an acrylate that when you dissolve this in the presence of cells and undergo a polymerization reaction, these end groups will link up and you'll create cross-linked hydrogel structures in situ, encapsulating cells. Now, I want to give a little bit of motivation to tell you why some of these just simple material structures are really useful for understanding matrix signaling and with cells. And the example I want to start with is heart valve fibrosis. So my group has had a long-standing interest in how you could engineer and grow heart valves. So for those of you that aren't familiar with heart valves, we have 4 valves in our heart, and they serve to direct flow from one chamber to the other. Now they're a perfect one-way valve that's designed to last our lifetime. So you can imagine opening and closing 60 to 80 times a minute, the pressure drop that goes across those valves. And it's really a perfect one-way valve. But sometimes these valves, so here's a picture of an aortic valve that's opening to allow blood flow, they can become diseased and fibrotic. And when that happens, the valves do not open and close properly anymore. And to date, there aren't any drugs that are known to reverse valve disease. So instead, you would have a replacement. So I had one student who was very interested in trying to understand the progression of disease and the cellular basis for this. And so I'll just show you a little example here that if you were to take one of these valve leaflets and section it into a thin section and then stain it so that you could see the cells and look at characteristics of the cells, one of the things that we know about these cells, so the cells are all these blue dots here, and the red is one of the proteins that they can make called alpha smooth muscle actin. It happens to be a protein that's really important in wound healing and helps the valve maintain its proper function. But when you get diseased valves, what happens is a lot of the cells get permanently turned on and express this protein, and they're turned on in wound healing. And this causes them to make lots of matrix and become very fibrotic and stiff. So we were interested in trying to understand what was the basis for causing some of this. And I was jointly advising a molecular, cellular, and developmental biology student on this project. And she was really interested in how this molecule, transforming growth factor beta, could be really important in making these VICs, these activated myofibroblasts that express this alpha smooth muscle actin. And so I'm just going to show you a little pictures. Normally, biologists, when they're culturing these heart valve cells, they'd take one of those leaflets, they'd digest away the matrix, and they'd put the cells on tissue culture plasticware. It's polystyrene. It's a really nice system for culturing cells so you can observe them under a microscope. But hopefully what I'll point out is that sometimes context matters in culturing these cells and how biomaterial systems can be useful. So Sharon, the student, was very interested in understanding the role of TGF-beta signaling in causing this. And hopefully you can see that when you add TGF-beta to these cultures, you see a huge amount of these myofibroblasts and their activation turn on. So Sharon was going to understand what's the difference over in this population versus this population with TGF-beta. But she did a simple control experiment. And the control experiment that she did was, she said, well, before I figure out what time points to look at this TGF-beta signaling, the first thing I want to do is just look at cells that I've freshly isolated from the valve, but I never let them touch tissue culture plasticware. And then another population that I put on tissue culture plasticware, and eventually I'm going to treat them with TGF-beta and see what happens and what time points to look at. Unfortunately, her time 0 of the fresh isolate, what she found, she did a gene expression profile looking at all the differences between these cells, and she found profound differences when you took cells that had never seen plastic and those that had been plated and just touched the surface for 12 hours. And she did a hierarchical clustering of these genes, and I'm not going to tell you all the details today, but hopefully you can see there's very large differences here. And I want to show you how large those differences are. By taking these valve cells and putting them on plasticware, just touching that surface, thousands of genes go up and thousands of genes go down. But now let me put that in context. So remember that TGF-beta in that image I showed you, how profoundly some of the myofibroblast properties are? Here's what happens when you treat those cells with transforming growth factor beta. That tens of genes go up. So in 8 hours and 24 hours, and only tens of genes go down. So in Sharon's experiments, this background of the material environment, putting them on a stiff plastic environment, confounds trying to understand this TGF-beta signaling. And note that a lot of these genes don't even overlap. So the material environment matters. And we've shown in a really simple sense that just by putting these cells in a softer environment that more resembles native valve tissue, and we looked at just a few key genes that these cells express, that here's the cells that you take out of the valve, but never plate them. Here's if you don't even take them out of the valve and look at the genes they express. And here's what happens if you just put them in a soft gel environment. It's not perfect, but it becomes a lot closer to the native environment than if you put them on this plastic plate where the expression profiles are completely different. So with that motivation, what I wanted to say, without getting into too much molecular biology, is that culture environment can have a profound difference on trying to understand cells and control their behavior. And so when we began to think about culturing cells and manipulating their properties, we think that matrix interactions really matter. And these can be physical interactions, these can be chemical interactions. So what I'd like to tell you is some of our recent efforts and how we've tried to make these matrices dynamic so that you can change from one type of environment to another in situ in the presence of the cells, and then understand how that cue influences those cells and matrix signaling. So the first thing I want to tell you about is how we think about this. And one is that sometimes we want to be experimenters and watch our cells in these hydrogel environments and then dynamically in situ change those material properties while we're watching it. So we dictate the change. Other times, we like to be passive observers and design systems that better mimic how cells are in their native environment, and we just observe and watch this. So let's look first at these user-dictated systems. So about 4 or 5 years ago, we got really interested in designing materials that could be dynamically changed, have dynamic changes in their property with light. And we developed this little monomer here that's a photolabile monomer. It has this nitrobenzyl ether group that when you expose it to light of selected wavelengths, it undergoes a cleavage reaction here. And so what we did was we made this monomer molecule and then we just coupled it to our polyethylene glycol molecule. So now what happens is when we react this molecule with various types of pegs of different functionalities, we create hydrogel networks. But now what's different is in these molecular meshes that are in this cross-linking density of the gel, these photolabile linkers can be cleaved by exposure to light. And when we cleave them with light, we have dynamic changes within the hydrogel. And this can all be done in the presence of cells. In fact, we chose this nitrobenzyl ether group because it was used as a caging molecule that would be delivered intracellularly and then exposing to light and releasing molecules and looking at internal cell signaling. So that's why we chose that particular group. But if we want to be able to make these changes and have user-dictated changes in these environments, the first thing we had to do is make sure that we could change the material properties and change them in a predictable and controllable manner. So what we did was we synthesized different gels, and then we put them on a rheometer. So just a little device that can shear these hydrogels. And it just so happens that the modulus of this gel is very, very sensitive to how cross-linked it is. And so this rheometer happens to have the ability to introduce a light source. And when you introduce that light source, it should cause cleavage and changes in this cross-linking density. And indeed, that's what happens. If you monitor this modulus of elasticity as a function of light exposure time, you see we start with a stiff gel. It's very cross-linked. And then when we expose to 20 milliwatts per centimeter squared of 365 nanometer light, the cross-links are cleaved and the modulus goes down with time. And we can fit that to an exponential fit, get a time constant of about 140 seconds. And when we half the light intensity, we double the time constant. And when we change the wavelength of the light to 405 nanometer light, the molecule absorbs that light differently. And we see a shift in the time constant that basically scales with the absorbance of the molecule at those wavelengths. So this all makes sense, and for those of you that know some photochemistry, this time constant exactly scales as one would expect, with the inverse of the light intensity and the inverse of the molar extinction coefficient. So that's all good. We can define these kinetics and understand how they cleave. But now the interesting experiment is, rather than continuously exposing those materials to light, now we can make materials that have a certain elasticity, and when you expose it to light, you can soften it. And then when you remove the light source, your material is now stable, but it's softened at a new steady state. And then you can expose again and soften again to a quarter of its original modulus. And so now we have materials that we can begin around a cell to change how it senses mechanical signals. And I want to tell you one of the new results that we've had in the way that we've used this material. So we have a long-standing interest in these bone marrow stem cells. So these mesenchymal stem cells are found in your bone marrow. They're an adult stem cell population. And many people are interested in using these as a source for different types of primary cells that are found in tissues, and using them to regenerate and regrow fat, cartilage, bone, muscle. And to date, there's a lot known about how I can take these multipotent cells and treat them with different factors in the media, insulin, transforming growth factor beta, to push them down different pathways of chondrocytes for chondrogenesis, dexamethasone or bone morphogenic proteins, take them down an osteogenic pathway towards bone. But what I'd like to say, though, is that while lots is known about dosing patterns and how to add things to media to control stem cell differentiation, I think there's also a growing appreciation that context matters. Just like culturing those valve cells on tissue culture plasticware has a profound effect on them, even when you're treating them with TGF-beta, we're starting to learn that these bone marrow stem cells are also profoundly influenced by their microenvironment. And one of those is also related to stiffness of that hydrogel. And without adding any molecules to the media, when cells sense different stiffness substrates, it can influence their fate. Very soft gels, making them become more like neurons. Intermediate gels, making them become more like muscle cells. Osteogenic gels and osteogenesis occurs on very stiff gels. And there are many different factors that people are interested in looking at. So what were we interested in? Well, when you make dynamic changes in gels, a couple of things. When we expose for different amounts of time by having a gradient in light exposure, we can make gels that have different modulus depending upon the position along here. If we move a plate across here, creating a gradient in light time exposure. And then we can measure that with a little probe, a nanoindenter, to get the local stiffness. This red curve is what we would predict from our rheometry data. The question that we were interested, though, is that if I dynamically change these environments, what would happen if I started seeding cells in stiff environments and then moved them to soft environments? And so, for example, these types of ranges are ranges that we can easily achieve with our hydrogel systems. And our question was, just like you dose cells with soluble factors, is there a dose effect of mechanical memory that I need to treat cells with high stiffnesses or soft stiffnesses? And is there a time that cells remember? And if they do remember this, is it ever reversible? Because as I culture and passage cells and treat them in different ways, this can be really important. And I'll give you one example of this. So again, many times with culture of these stem cells, we first passage cells on this plasticware. And then we might start our experiment once we get a large number of cells and put them in hydrogel environments. Well, what's interesting here is that depending upon how long they touch super physiological stiff tissue culture plasticware, which is about a gigapascal, so whether it's a day or 7 days, what happens is you start to change things in these stem cells, and they start to express transcription factors in their nucleus that are associated with them becoming bone. And then we could also correlate this to another transcription factor that's starting to be implicated as how it senses that mechanics. That part's not so important. Let's just look at this RunX2. And so the question, though, is that if I seed cells on tissue culture plasticware and then I put them in a soft environment, unfortunately, what happens is the longer they've been on this stiff environment, the more they express these osteogenic genes, even after I put it on this soft environment. So increased stiffness causes osteogenic genes to be upregulated, and it has a profound effect on what happens to your experiment here. Let me show you how we want to use our in situ gels for this. If we culture cells in stiff environments, then I can in situ soften them and watch how they respond to this. So much like I walked you through, cells in a stiff environment, soften, soften, soften. And so one of the things that we showed, and let's just look here at this bottom one, is that in the soft environment, this osteoblast transcription factor is very low. And up here in this regions, it's very high. So the interesting question is what happens if you seed cells in stiff environments and then soften them at different points in time? Can the cells switch between these activated and inactivated states? So here's the experiment. We're going to culture cells just for 1 day on these stiff environments and then soften. And let's look just here. The stiff environment, they express this osteogenic transcription factor. And then if we soften it, what happens is it reverses back, and that goes away from the nucleus. It goes back and behaves like it's in a soft environment, not becoming an osteoblast. But now let's look here. What happens if you let it be in an environment for 7 days and then soften it? So look down here at 7 days. In the stiff environment, it expresses that osteogenic transcription factor. And then when I soften, it goes down and reverses again. It just takes a little bit longer time. But now the last point, what happens if it's 10 days? 10 days in a stiff environment, it expresses this osteogenic transcription factor. But when I soften it, even after 10 days after softening, it appears to be irreversible. Something changed inside the cell nucleus that it no longer can respond to its new environment, and it's set on this pathway of becoming perhaps an osteoblast. So the reason I think that some of this is important is that we can begin to think about when you culture cells, there can be unintended consequences. So if you're just passaging cells before you're starting your experiment, can cause irreversible changes in cells. And the second is we can learn about designing our biomaterial niches and think about extracellular matrix cues as giving a critical dose time, so we can learn about when cells get that signal reversibly and they can switch back and forth, versus when it becomes irreversibly if I'm trying to push cells down a pathway to make certain cell types. Okay, so this is about dynamic changes in the gel, but the other nice thing about spatial changes in the gel is I can also begin to learn about how are cells sensing this. So I observed this change with mechanical properties on cell fate, but what I'd really like to know is how does the cell sense that? And with photochemistry, you have spatial control of gel properties, because I can come in with a laser beam and modify around these stem cells their ability to attach to the underlying hydrogel properly. So here I'm just showing one example where we erode away some of the gel so the cell can't attach here. You can also cleave adhesion molecules. And let me show you a little movie here. So we're watching these stem cells on our stiff environment. And when I click on this, we're going to erode away some of the underlying gel. So it's well attached. We've just eroded this adhesive site. And the cell dynamically contracts and reorganizes its cytoskeleton. Let's look at that. So we erode this away. The cell's pulling on this. There's a force. And that force is changing things in the nucleus. So we use this type of experiment and information to get a better understanding of what those forces are. And we can characterize things like the time lag of how long it took the cell to respond, the time constant for the remodeling and the retraction. And so when we model this with things like viscoelasticity and viscoelastic retraction that we often use with polymer systems, we can learn about these time constants and compare how they're much different and have different induction times and retraction times than if they're on stiff tissue culture polystyrene or glass or other types of modulus. And we're beginning to correlate what is the mechanosensor and what is actually linking all the way from the extracellular environment to the nucleus. Okay, so everything I've told you about so far, though, is with mechanical signaling to cells. But chemical signals clearly matter as well. And they can synergize with mechanical signals. So what happens if I give a chemical ligand or signal to a stem cell, and try to take it away or introduce it? And what influence might that have on cell fate? So the example I'm going to show you here is we take our same monomer and we can attach any amine-containing molecule. This happens to be an amino acid sequence of arginine, glycine, aspartic acid that we couple to our photolabile monomer. And this is one that can bind to integrins on cells, and it's found in fibronectin, an adhesive protein. But now what we can do is we can introduce it as a pendant functionality that can be cleaved upon exposure to light. So here's the experiment we were interested in. We have lots of interest in controlling these stem cell, bone marrow stem cells, these MSCs, and pushing them down differentiation pathways. And one that we're interested in is chondrogenesis. Now the thing about these MSCs is that when they're becoming chondrocytes, many, many things happen. And I'm going to simplify just a few things. So one is early on is they make this adhesive protein fibronectin. And this causes the chondrocytes to aggregate and form these lacunae. And that causes different intracellular signaling cascades important to chondrogenesis. And one of those causes deposition of these proteoglycans, these glycosaminoglycans that I showed you yesterday. And they downregulate fibronectin. They're building up this cartilage-like matrix environment. And they begin to form a richer paracellular matrix by about 2 weeks along this process. Now the thing about these MSCs is if you just culture them in hydrogel environments without any adhesive ligands such as RGD, they'll die because of lack of matrix interactions. They undergo anoecus. So without an adhesive ligand, after 4 days, half of your cells will die off because they require adhesion. They're in a bone marrow niche where they're well attached. So if you add RGD, you can culture these cells and maintain high levels of viability. And many biomaterial scientists often put RGD into hydrogel matrices to promote cell survival. It's a very common approach. But the question we ask is that these cells normally like to have the ligand around early, but not later, as they're building up this lacunae structure and spherical cell morphology. So our idea is, well, what would happen if you put in this RGD to allow survival, but cleave it at different times? Because we were hoping that this would further promote chondrogenesis, and that its persistence, we were thinking or hypothesizing, might prevent chondrogenesis. And so that's the experiment. You could cleave this at different times, but I'm just going to show you the data, what happens when you cleave it at day 10. So what happens here, if I look at this GAG production, one of the early markers of becoming a chondrocyte as a function of time, early on, so in the PEG hydrogels, it's very low, and it's very low because the cells are dying. They can't survive in these gels. But when RGD is present, it's also very low, but the cells are surviving. They're just not making a lot of this molecule. And then in the blue line, if we cleave the RGD at day 10, what happens is we begin to see an increase in the production of the GAG. So that's one measure that perhaps we're helping them become cartilage cells and chondrocytes. And we can look at this more directly because one of the cell surface ligands, the integrins that binds to RGD, is alpha V beta 3. And we have a stain, so we can look at that. And when we look at that, early on, at day 4, almost all the cells are positive for that integrin. They're interacting with our RGD. And when the RGD persists and remains in the gel for the whole experiment over 3 weeks, almost all the cells are positive for that. But when we cleave it at day 10 and then look 3 weeks later, we see very, very few cells positive for that integrin. So removal of that RGD influences the integrin expression on the cell. So an evidence that they're interacting with our signal. And then evidence that it influences chondrogenesis. We can stain for one marker, CD105, that's a stem cell. And we can also stain for collagen 2, which is a later stage of a cell becoming a chondrocyte. And what happens early on is that almost all the cells have this stem cell marker. But when RGD persists, some of the cells still express the chondrogenic marker. They're in this hydrogel that has elasticity like cartilage, and it forces the spherical morphology. So we get some chondrogenesis. About half of them still have the stem cell marker. But by 3 weeks, we can get many more cells expressing this collagen, this chondrocyte marker, and fewer with the stem cell marker. So this is one signal, one small change that you can begin to enrich populations and make more chondrocytes from this stem cell population. So these were all examples of physical and biochemical cues and dynamic changes that, as an experimenter, I could come in with light and change the signals around a cell. But other times, I want to be an observer, and I just want to make a material environment that cells behave much more like they would in a native tissue system. And yesterday, I talked a little bit about how we could make systems and our approach to designing ones that cells could bind to, degrade, encounter different signals, all without me changing the environment, but the cell deciding and making proteases to remodel this local system. And we used the native extracellular matrix as a guide to figure out how we wanted to design our synthetic material systems. And so where our synthetic hydrogels and our photolytical ones gave mechanical cues and high water content, and we could put in peptides that would mimic adhesive proteins, the big difference was that our system was light degradable, and instead we want a cell degradable, when they make enzymes that they can cleave and remodel this. And so we've been really interested in designing different types of peptides that can cause adhesion, deliver different types of growth factors. This is one that binds that TGF-beta that's really important in many different cell processes. But this sequence here is one that cells can cleave and degrade and is found in collagen. So to make these, this gets to the question of how can I make different types of systems that take advantage of orthogonal chemistries. And so we were making our PEG hydrogels, and we wanted to take advantage of one of these click chemistries, these orthogonal chemistries. And at the time we were doing this work, Carolyn Bertozzi's group had recently published on this cyclooctane that could react with this azide to form this triazole. And the big advantage here was that this click chemistry of alkynes and azide was copper catalyzed. And copper is very toxic to cells. So Carolyn's work showed that this cyclooctane that was difluorinated could react under aqueous conditions at 37 degrees C without the copper catalyst. So we made molecules with this cyclooctane that's difluorinated, and I'll tell you it's a very, very difficult synthesis. It required lots of effort, and right now you can use lots of other more easily accessible click chemistries, but when we began our work, this was one of the few that was cytocompatible. And we made our peptide sequences with these azide functionalities containing this cell cleavable group. So when you put cells inside of these environments, you can click together different physical properties from your PEG and different biological signals from your peptide and look for different unique properties that you'd like to have if it's a stem cell you're trying to differentiate or whether you're trying to look at cells moving or proliferating. And what I'll show you is that when you mix these two together and monitor now with the rheometer to monitor gel formation, so we're looking at the modulus as a function of time, you'll see that here the evolution of the gel takes tens of minutes, but there's no copper required, and the gel point, crossover's estimate of the gel point was about 5 minutes. So you can form these in the presence of cells, but the important thing here is now I'm the observer. Let's watch these cells, and as I watch these cells inside of these gels, they can spread and attach with the RGD ligand, and they can degrade and remodel this matrix. So the cells are deciding to degrade and make tracks through here. So now I'm the observer. So what's this good for? Well, we are interested in a question and had a collaboration where we were trying to understand how cancer cells might metastasize through a matrix, how this might depend upon their ability and mechanism to remodel the matrix environment. So cancer cells can grow into small tumors, and one of the things that matters a lot with these tumors is many times you feel them because they become stiff. Matrix environment matters. But what you really care about is if this tumor stays together, I can excise it. But sometimes what happens is you'll get cells that will break away. And what caused that cell to break away? And for it to be able to break away, it has to be able to locally degrade this matrix environment. What changes caused that? And clearly the cell seems to be having trailing cells. So what happens to that lead cell? Because when metastasis occurs, cancer becomes very difficult to treat. So we were interested in by what mechanism does that leading cell and different cells remodel and change that environment. And the things that can happen is sometimes people think that the cells are degrading that matrix, and it's called a mesenchymal type of migration, where they attach and degrade, kind of like that little movie that I showed you. But other times, cancer cells are thought to be able to squeeze through and deform through really tiny pores, and have a very amoeboid mechanism, where some of the matrix remodeling and matrix interactions don't matter so much. So it's really important to know which of these mechanisms occur, because that can help you figure out what types of drugs to use to stop this. So let me just show you one of the things we learned from this was we cultured these cancer cells that came from a patient that had metastatic melanoma, and we were interested in which of these mechanisms occurred. And we changed our gel, so this is one that the cells can easily degrade and cleave, and it looks like the cells are degrading and spreading through this mesenchymal mechanism. So then that might be a good way or good target to block some of that proteolysis. But unfortunately, we learned something that when you change this, and misspell and put, instead of this tryptophan, this alanine, watch what happens here. The same cells transform their mechanism, and they can squeeze through tiny little pores and be more amoeboid. So that's one of the problems. These cells become very plastic. So what we're interested in trying to figure out are how do these cells become plastic, and what are some of the differences in this leading cell? Could I culture this? Could I give it a signal here that could facilitate this and capture that cell so that I could analyze it and figure out molecular level changes? So this brings me to my very last part of my talk, where we went through examples of user-dictated changes in hydrogels, one where I'm an observer and watching cells in a native-like environment and letting the cells remodel. Now can I have cells in that native-like environment and dynamically change properties around it and introduce different cues? So let cells introduce a cue so cells can attach, and then let cells degrade it. Or perhaps introduce this cue and remove it and put in a different type of cue, or allow two cells to interact. So we're really interested in more complex types of environment, and this brings us to having sequential and orthogonal reactions. And that's why we picked this first click reaction, because we have a second click reaction in hand that I talked about yesterday, this thiol-ene click reaction, where we could react enes with thiols, and this is a photo-initiated conjugation reaction that we can use. And the nice thing about this is that when we make our peptides, one of the protecting groups on the peptides to prevent branching and side reactions is this allo-oc group, this ene, and I can take advantage of that ene and react various thiols. So let me show you this pictorially. So pictorially, what happens is we can introduce thiol molecules, swell them into our gel, expose it to light of different dosages in the presence of different initiating molecules, and we can titrate in different amounts of this signal and conjugating it in a way that has concentrations of biological relevance. So I can couple those to my matrix, and I can couple them in a way that's spatially defined. So if I put a mask over this, only where the light is exposed do I conjugate the molecule. So I can spatially define this. And then if I use fancier multi-photon pattern with a confocal microscope, I can pattern chemistries in 3D. So this means I can culture cells, watch them, and introduce a ligand to the cell and watch how they would respond. And this is all done just in real time. It takes seconds to do this. But I can introduce a cue, but we also have this photolabile moiety that I told you about. So introducing is one thing, but I also want to be able to remove it. And if I put in that photolabile linker, I can couple in molecules, and then I can cleave them and remove them and take them away by cleaving crosslinks in my network. But the tricky part here is I have to have independence of light of those two. And so our cleavage reaction occurs. I showed you examples of 365 nanometer light, and it can go all the way out to 405. And if I use a molecule for my thiol-ene reaction, then I can couple at longer wavelengths that are independent of my cleavage. So that means I can write chemistry in this wavelength, and I can take away chemistry in this wavelength. And that's what I'm showing here, is the amount of peptide that will stay intact after I conjugate it when I expose to the long visible wavelengths. And then over here, I can cleave and take it away. So let's look at a picture of that. So we can add in chemistry with the long wavelengths, and now cleave by exposure to that 365, and I can dynamically change patterns. I can remove certain signals, such as the red signal here. So why might this be useful? Well, I can now click in chemistry and make hydrogels of cells encapsulated in a 3D environment. I can add in signals to change local signals in that 3D environment. maybe add in a second signal. So within a single material environment, I create no signal, the red signal, the green signal, and combined signals. So I have a rapid way of looking at how various signals can influence cells. And then I can either degrade this region and remove that signal, or I could even degrade and capture those cells, much like I wanted to capture that cancer cell. And then perhaps at a later time, remove these. And it's all cytocompatible, so I can capture these cells, culture them, expand them, do further experiments on the system. Perhaps for my tissue engineering applications, I began to now think about ways where I can guide cells and direct them in 3 dimensions, where I can erode channels, but cells will not go down these channels unless there's the proper adhesive ligand allowing them to move. So I can direct a collection of cells to move in certain directions, perhaps to innervate a muscle, or perhaps to grow a blood vessel. And this can all be done in complex 3-dimensional manner, directing and guiding cells, even in the context or presence of other cells that might exist in the bulk. So this is very much futuristic, but the possibilities of where things could go in 3-dimensional cell culture. So in this talk, I tried to blend advances in chemistry with more advanced biological questions, and I hope that you might appreciate how this could be used to tackle and understand tissue engineering applications and current bottlenecks. So there are lots of different cytocompatible chemistries that allow for the synthesis of very versatile 3D cell culture platforms. I think what's unique is that we can use dynamic changes in material properties and dynamic introduction of functionalities to answer important biological questions, such as mechanical memory effects on stem cells and how that influences their pluripotency. How temporal regulations of signals could influence the ability to differentiate cells and to enrich a population of chondrocytes. Orthogonal chemistries, being able to do multiple sequences. Cells get multiple signals that are involved in wound healing and as a stem cell migrates from its niche to repair a tissue. And orthogonal chemistries are helping us begin to develop advanced culture systems and answer more sophisticated questions. So in conclusion, I also want to make sure to acknowledge a great group of students that I've had the opportunity to mentor and advise, both past and present, at the University of Colorado. The particular work that I talked about today is a collaboration with Leslie Lindwan and Dr. Sharon Wong, who recently graduated, on the heart valve project. On the MSCs and their chondrogenic differentiation and their memory, that was April Cloxon developed all the photodegradable work, along with Mark Tibbett. And Mark Tibbett and Chun Yang did all the work with the MSC memory. And then the last bit with the cancer cells and the multiple orthogonal reactions, Natalie Ahn and Mike Schwartz did collaborate with us on the cancer work, and Cole DeForest did all the work with the orthogonal types of reactions and advanced patterning. So with that, I'd be delighted to answer questions that you might have.